everyone. In this lecture, I'll talk about some steps of the downstream processing of the fermentation process. Mainly, I'll talk about the harvesting of the cells and disruption of the cells. Okay, so first harvesting the cells. Okay, so let's say harvesting of the microbial cells. Okay, so now, I mean, usually the equipments which are used to harvest the cells, they are same for both native microbial cells or recombinant cells. However, I mean, in case of recombinant cells, there is some physiological changes. I mean, the recombinant microbial cells, they are physiologically different than the native microbial cells. So that's why the conditions which are optimal to harvest the native microbial cells may not work. May not work like the same in case of for recombinant microbial cells. Okay, so that's why sometimes modification is necessary when we want to harvest the recombinant microbial cells. Okay, and there are different methods to harvest or separate the microbial cells from the liquid part. Okay, so such as high speed centrifugation, high speed semi continuous or continuous centrifugation, uh, membrane microfiltration. So how about like the if we just say that high speed centrifugation. Okay, and the centrifuges that are used in the industry are different than the centrifuge machines that we use in our laboratory. Okay, because the centrifuges that we use in our laboratory usually have less capacity. I mean, they cannot handle too many liquids. Okay, but in the industry, the fermentation, I mean, we do fermentation in very big bioreactor. That means there are lots of liquids. Okay, so in that case, a special type of centrifuge machines are required. Okay, usually in industry, semi-continuous or continuous high-speed centrifuges are used. Okay, so let's talk about the high-speed semi-continuous centrifugation. Okay, the in this case, I mean, in for this centrifuge machines, cell suspension is continuously fed into a running centrifuge. Okay, the running centrifuge and the cells are concentrated within it, while the clarified medium is collected in an external container. Okay, and when the centrifuge chamber is full of packed cell, we have to stop it and need to remove the cells. However, I mean, this kind of centrifugation process, the semi-continuous high-speed centrifugation process, it has some disadvantages. Why? I mean, sometimes it need to be stopped and then restart the procedures. Why it need to be stopped? To harvest the cells. Okay, and especially when large volumes are being processed, this stopping and then restarting can be a major inconvenience. Okay, uh, and the cost of both the equipment and the power to run is high, and the release of the microorganisms into the air as aerosols is possible. Okay, and then if it occurs, it will contaminate the environment. And finally, and then the difficulty in removing all the microbial cells from the spent medium. Okay, I mean, when we use semi-continuous centrifugation, sometimes it's difficult to separate all the microbial cells from the media. Okay, so and in the industry there are, I mean, different types of semi-continuous or continuous centrifugation machine. So I'll suggest you to Google it. In that case, you will see that there are different design, or the design of centrifuge machines, and the mechanisms is different in these machines. In the slide, I give an example figure of a continuous. I mean, it's a high speed centrifuge machines, continuous high speed centrifuge machines. Okay, so if you look at it, its mechanism is like this. So uh, we can continuously feed the flow of the spent media. This media, I mean, this spent media contains the cells. Okay, Then this flow, it goes these directions, and the centrifugation occurs, and because of the centrifugal force, the cells, they get deposited in this side. Okay, and the media, the liquid portion of this media, they overflow. It's natural, right? If the cells, they get deposited here, definitely the liquid part of the this feed will be will get out, okay, from the outlet. Okay, so this is a continuous type centrifugation. Why? How we can achieve this continuous working? Like, there are some provisions by using. There are some out points. From those out points, we can collect those solids. Okay, those uh, solids. Which has been deposited here because of the centrifugal force. Okay, so usually in the industry, this kind of continuous, uh, high speed continuous centrifuge machines are used because these are efficient, right? So if we try to use the benchtop type centrifuge, or the centrifuge that we used in the lab, it will not be it will not be that efficient or convenient for the industry, right? Because if we want to use those kind of centrifugation machine, we have to centrifuge the we have to centrifuge in a batch way. Batch way means first we centrifuge some media, then some other media, then other. Okay, batch wise. But doing the separation by in batch wise centrifugation is not a efficient or convenient way for the industry, because the fermentation industry, they have to deal with thousands of millions of liters of liquid. Okay, so this was about centrifugation, and another methods to separate the cells from the media is membrane filtration. Okay, so regarding the membrane filtration, there are two methods. Okay, one is traditional filtration, which is also called dead in filtration. Another is cross flow filtration. So what is the traditional dead in filtration? It's like this. Okay, I mean the machines where we will do the separation, it have the membrane filter, and we will flow our spent media, which contains the cells from the above, right? So these cells, as their size is bigger than the pore size of the membrane, the cells will stuck on top of the membrane, and the liquid will pass out. Okay, pass through, and then we can collect this liquid. So by this way, we can separate the cells from the liquid. Now, this traditional filtration method has problem, because as these cells, they they are deposited here. Soon the flow rate decrease. Okay, and the I mean the and the flow rate decrease. Okay, and sometimes clogging of the cells can occur here. However, if we uh, increase the pressure, pressure of media flow, then maybe the flow rate of the media can be increased, but yet soon it becomes actually clogged. Okay, and the separation stops. Okay, however, I mean if we use cross flow filtration system instead of traditional dead end filtration system, we can overcome this problem to some extent. Okay, so what we understand by cross flow filtration? Okay, in this case, passing the cell suspension at a high speed across the surface of the membrane. Okay, the cell suspension, that means media with the cells, they are uh, passes across. Not like this way. Okay, from the top, rather side by side. Okay, in a cross flow direction. Okay, so what happens under these conditions? Only a very small fraction of the circulating liquid goes through the membrane in one pass. Okay, it's the liquid they keep recirculating. Okay, so uh, what happens? Due to the force that this circulation of the liquid creates, they sweep away some of the cells. Okay, they sweep some of the deposited cells from here. Okay, so that's why this kind of filters, they don't get clogged as rapidly like the traditional dead end type filtration. Okay. Okay, and usually the filters that are used for filtration, their average pore size is like from 0 to 0.45 micrometer. Okay, and after many cycles in a cross flow filtration system, almost all of the culture medium will have passed through the membrane because we just recirculate them. Okay, and the use of cross flow filtration is generally limited to small scale operations. Okay, and because although this this kind of cross flow filtration, they are mainly used in a small scale industry, because although it has some benefit over the traditional dead end filtration, yet the filters, they get clogged. Okay, they actually, all the getting of clogged may delay, but they eventually get clogged. Okay, so so that's why till now centrifugation is most widely used in the large scale industry. Okay, not the filtration system. Okay, now after you harvested the cells, after we separated the cell from the media. So if our target product is in the media, then we'll work with the media. We then have to purify our product and package them and send them to the market to sell. But if our target product is in the cell, okay, in that case, we have to disrupt the cells to harvest our products. Okay, and the procedures, I mean, what we understand by disrupting the cells, to break it down. Okay, so the procedures that are used for disrupting the cell must be vigorous enough to break the microbial cell walls, yet gentle enough to ensure that the protein product is not denatured. Okay, this is a very important point. We can vigorously break the cells. The more vigorous the procedures will be, the cells will be, we can break the cells more easily. But we cannot just simply do that. We cannot just simply use lots of forces, because if the forces are too much, they will break the cells, but at the same time they can break the protein product, our target product. Okay, so that's why the procedures that will be used should be vigorous enough to break the cell wall, but not the protein product. Okay, our target or desired protein product. Okay, and cell disruption can be done by chemical, biological, and physical methods. Okay, however, there is no universal method of cell disruption, or you can say there is no single set of conditions for cell wall lysis, because cell wall of different microorganisms are different. We know that, right? Say for example, cell wall of the gram positive bacteria, gram negative bacteria, yeast, they are different. Okay, and as the cell wall composition of the microorganisms are different, the strength of the cell wall is different because of the chemical composition, because cell wall of different microbes are made of different polymers. Okay, so as the strength of the cell wall is different, the process that we will use to disrupt the cells will be different. Okay, so now the strength of the cell wall composition, the strength of the cell, okay, and the composition of the cell wall, it depends on several factors such as the culture conditions, the cellular growth rate, the phase of the growth cycle when the cells are harvested. What this means, the cell wall strength of the exponentially growth phase cells or the stationary phase cells have different strength. Okay, then how the concentrated cells are stored. Okay, because storing has some effects. Okay, whether the isolated microorganisms was expressing a clone gene, because we already learned this thing, right? The cell wall of the cloned, the recombinant microbes and native microbes, they are physiologically different. Okay, so if we are using the recombinant microbes, okay, and based on the gene that was cloned into that recombinant microbes, the cell wall composition can be different than the native one. Okay, so all these factors affect the cell's susceptibility for disruption, because these factors affect the cell wall composition and strength of the cell wall. Okay, so now let's talk about the methods of cell disruption. First, chemical methods. Okay, and we can disrupt the cells by treating with alkali organic solvents or detergents. Okay, so which one we will use? Which we will use alkali or organic solvents or detergents? Which one we will use depend on the types of the cells and our target product. Okay, so such as, for example, if the protein product is stable at pH 10.5 to 12.5. Okay, in that case, bacterial cell lysis can easily be done at low cost, such as, for example, RHGS. It means recombinant human growth hormone is efficiently released from E. coli by treatment with NaCl, sodium chloride, at pH 11. Okay, I mean, this product RHGS, they are stable in high pH. Okay, and actually, if our cells are treated at this pH, they easily release the product. The cells get easily lysed and they release their intracellular product. Okay. Now it was about using the salt. Now, treatment with an organic solvent is a simple and inexpensive way to disrupt cells and has been used for the isolation of enzymes from yeasts. Okay. And detergents, when we use detergents, what detergents do? Detergents permeabilize bacterial cells by solubilizing cell membranes and membrane proteins. It makes holes and pores in the cell wall, and thus the proteins and other molecules, they can escape or get released from the cells. If we treat the cells with detergents, they make hole or pore in the cell wall and cell membrane of the microbe. So the intracellular protein or other molecules, they can get released and get out from the cells through this pores or holes. Okay. However, detergents are expensive. Okay, and uh, they can they can frequently denature the protein product and are often retained as contaminants throughout the purification process. Okay, the residual effects of detergent is very high. Okay, they are expensive and they can denature the our target protein product. Okay, so these were about the chemical methods for disrupting the cells. Now the biological methods to disrupt the cell. Okay, the major biological method for disrupting microbial cells is enzymatic lysis. For example, the cell walls of gram-positive bacteria, they can be lysed by lysozyme. Okay, it's an enzyme. Okay, and the walls of gram-negative bacteria are lysed, or the lysis is done by hydrolysis. Okay, are hydrolyzed by the enzyme lysozyme, and metal chelating agent EDT. Okay, EDT is a chemical. Okay, EDT is very widely used. Okay, also not only in the industry, also in the laboratory. Okay, EDT is frequently used to disrupt the or lyse the cell walls of bacteria. Okay, and the cell walls of yeast are hydrolyzed by combinations of one or more of the enzymes. These enzymes like 1,3 glucanase, 1,6 glucanase, mannase, and chitinase. Cell wall of yeast is made of chitin, so that's why we have to use these enzymes, chitinase. Okay, it's necessary. Chitin is different. It's a different type of polymer. The bacterial cell wall, they don't contain chitin. Okay, only yeast contain the chitin in the cell wall. The advantage of using biological methods are highly specific, and the conditions for lysis are mild. but disadvantages is costly, because in biological method we use enzymes, and enzymes are costly. Okay. Now, physical method. Okay, and physically, cell can be disrupted by non-mechanical methods or mechanical procedures. And non-mechanical methods are like osmotic shock, repeated cycles of freezing and thawing, that means freeze-thaw effect. And mechanical procedures are sonication, wet milling, high pressure homogenization, and impingement. Okay. However, after treatment by a non-mechanical method, many of the cells remain intact. Non-mechanical methods are there less efficient than the mechanical procedures. Okay, and the mechanical disruption they are highly efficient. Okay, so that's why mechanical disruption is more frequently used than the non-mechanical methods. Okay, so now, I mean, okay, we learned about the chemical methods, biological methods, and physical methods of disrupting the cells. I think in this slide there are some, terminology which we may not be familiar. So, like we know what osmotic shock is. Okay, we know what sonication is, but like wet milling, high pressure homogenization, and impingement, some of us may be not that familiar with the this process. So that's why in next few slides I'll talk about this process. Okay, this mechanical process of cell disruption. Okay, so first, wet milling. So what wet milling is? So like, what is done in the wet milling? A concentrated cell suspension is pumped into the chamber of a high-speed agitator bed mill. Okay, and this is the agitator bed mill, and it contains lots of small glass beads. Okay, and the diameter of glass beads are less than 1 millimeter. Okay, and there is a central shaft which we can rotate through a motor. Okay, and this central shaft has number of blades. Okay, you can see only two blades here, but it's not necessary that this shaft will contain only two blades. It can contain number of blades. Okay, so there are a small glass beads and also shaft with attached blades. So in this process we just have to fill this compartment with our media. Fill this compartment with the cells that we want to disrupt. Okay, then if then when the device is turned on and the blades are put in motion, if we turn on the motor, the blades will move. Okay, and then most of the cell disruption occurs as a consequence of the shear force generated by the high speed motion of the glass beads. When these blades will rotate, they will also cause these beads to move. Okay, this small glass beads, and this is this motion of the small glass beads. In what speed they will move will depend on the speed of the central shaft. Okay, so usually high speed is used, so that's why high speed motion is achieved by the glass beads. The glass beads move very fast. Okay, and this cause a great shear forces. Okay, and this shear force cause the disruption of the cells. Okay, you know nowadays in the laboratory, cell lysis is mainly done by chemical methods. In the laboratory, I'm not talking about the industry. In the laboratory, mainly done by chemical methods, because in laboratory using the chemical methods are faster and more precise. But previously, when this kind of chemical kits were not that much available, glass beads were used to use to disrupt the cells, to harvest plasmids, etc. Okay. Okay. Now the optimized cell disruption in by this process, that means wet milling, depends on several factors, and those are the number and centrifugation of the agitator disc. Okay, the agitator speed, the size and number of the glass beads, the concentration of the cell, the geometry of the grinding chamber, and the temperature. Okay, now high pressure homogenization. Okay, what I mean, how the cell get disrupted by this process. So I mean, think this is a high pressure homogenizer. I mean, the homogenization will be done inside it. So concentrated cells are pumped into the valve assembly under high pressure. I mean, the cells that we want to disrupt, first we need to concentrate them, and then we'll pump them here with very high pressure. Okay, the pressure will be very high inside. Then the pressure is rapidly decreased. I mean, the cells will be in very high pressure, and then the pressure will be rapidly decreased. So this rapid change in the tension will cause the cells to lyse. Okay, this process can be customized for different microorganisms and protein products. I mean, based on the microorganism and our target protein products, we can customize this process. I mean, what kind of pressure we'll use. Okay, how we'll decrease the pressure. Okay, and how we can customize this process by changing the operating pressure. Okay, and by design of the valve, the temperature of the cell suspension, and the number of times the cell mass is treated. Okay, I mean, it's not necessary that we treat the cells only once. Okay, we can repeat the process. Okay, we can I mean, treat the same cell mass for several times, because if we just treat the cell mass for only one times, many of the cells, they will not get disrupted. Okay, so to confirm that all the cells are disrupted, usually this process is repeated. Okay, then the last one, impingement. Okay, so look at this figure. I mean, cell suspension is pumped in direction and also in this direction. Okay, I mean, cell suspension, they are pumped in opposite directions. So what will happen eventually? They will collide, right? Okay, they will impinge with each other. So it will cause the disruption of the cells. This is the basic of the uh, of the impingement method. Okay, so what we can do? Okay, we can do this in another way. I mean, in this case, in this figure, the cells was I mean, pumped in opposite direction. Okay, but instead of this, we can just pump the we can just pump the cell suspension towards a stationary surface. Okay, so when the cells will hit the surface, they will get lyse. Okay, so a high velocity stream of suspended cells under pressure hits either a stationary surface or a second fluid stream of suspended cells. Okay, and the forces that are created at the point of contact will disrupt the cells. Okay, and with this device, a high percentage of the cells are disrupted by a single passage through the unit. Okay, and unlike high pressure homogenizers and high speed agitator bead mills, which generally require highly concentrated cell suspensions, this device can be used to disrupt cells in either dilute or concentrated preparations. I mean, this process, the wet mill and high pressure homogenization, if we want to disrupt our cells by this process, we need to use high concentrated cell. Okay, but if we want to use impingement method, either high concentrated cell mass or diluted cell mass is okay. Okay, and the activities of cellular proteins are not significantly impaired by the technique. Okay, that means this technique is good, better for our target protein. Okay, so these were some mechanical processes by which we can disrupt the cells. Then next, this slide summarize the upstream processes and downstream processes of a fermentation process. I think we all already know about the different upstream processes and downstream processes. So that's why I'm not going to discuss about this. Okay. So in the next, this is all for this lecture. In the next lecture, I'll talk about some major challenges that the industrial biotechnology is facing. Okay. Till then, be safe. Thank you.